Today I'm talking about the principle of clinical trial design, and since I have only 15 minutes, I'm going to explain only most important things. Okay, so this presentation is based on ICH E9 and E10, and I'm going to focus on only a confirmatory trial. Okay, so the most important thing in clinical trial is to minimize bias. Okay, so the term bias describes the systematic tendency of any factors associated with the design, conduct, analysis, and interpretation of the result of a clinical trial to make the estimate of a treatment deviate from its true value. So actually, if we have a significant result in clinical trial, there are four possible reasons. The first one is the data is wrong. For example, if we input data, we may make a mistake. So in order to prevent such mistake, we need clinical data management. So clinical data management is a very important issue. Second reason is bias. Okay, so in this presentation, I focus on to minimize bias. And third one is chance, and fourth one is new treatment is really effective. So the most important design techniques for avoiding bias in clinical trials are blinding and randomization. And these should be the normal features of most controlled clinical trials. So a double-blind trial is one in which neither the subject nor any of the investigator or sponsor staff involved in the treatment or clinical evaluation of subject are unaware of the treatment received. So blinding is intended to limit the occurrence of conscious and unconscious bias in the conduct of an interpretation of a clinical trial, arising from the influence that the knowledge of treatment may have on. So, if we don't have blinding, it may influence on these all things. The first one is recruitment of the allocation of subject, and next one is their subsequent care, and so on and so on. And next one is randomization. So randomization is to allocate patient via random assignment to a treatment or control. So randomization also tends to produce treatment group in which the distribution of the prognostic factors, known and even unknown, are similar. So in combination with blinding, randomization helps us avoid possible bias in the selection and allocation of subject. And also there are some other things which we can do in order to minimize bias. For example, we should pre-specify statistical analysis method, or we should analyze an appropriate analysis set, such as ITT set or PP set, and we should handle missing data with appropriate statistical method. Next issue is sample size and power. So the number of subjects in a clinical trial should always be large enough to provide a reliable answer to the questions addressed. So this number, which is also known as sample size, usually determined by the primary objective of trial. So conventionally, the probability of type 1 error is set at 5%, and probability of type 2 error is conventionally set at 10 or 20%. So the most simple analysis is two sample t-test. So in that case, sample size per group is given by this formula. And in the numerator, sigma square is the population variance, which is unknown value. Also in the denominator, delta is effect size, which is also unknown value. So we have to estimate. So in confirmatory trial, assumptions such as sigma square and effect size should be normally based on published data or on the result of the earlier trials. And the treatment difference to be detected, so this means the delta, may be based on judgment concerning the minimal effect which had the clinical relevance in the management of patient, or on a judgment concerning the anticipated effect of the new treatment, where this is larger. So if the sample size is not large enough, the clinical trial cannot produce a significant result, although actually the result is significant. That error is called the type 2 error in statistics. Next issue is control. So the choice of control group is always critical decision in designing a clinical trial. So that choice affects, first one is the inference that can be drawn from the trial. Second one is the ethical acceptability of the trial. And third one is the degree to which bias in conducting and analyzing the study can be minimized. Fourth one is the types of subject that can be recruited and the pace of the recruitment. And the fifth one is the kind of endpoint that can be studied. And so on and so on. So I'm not going to read all of them. So control group have one major purpose. That is to allow discrimination of patient outcomes. For example, change in symptoms, signs, and other morbidity caused by the test treatment from outcomes caused by other factors, such as natural progression of the disease, observer or patient expectations. So the control group experience tells us what would have happened to patient if they had not received the test treatment, or if they had received a different treatment known to be effective. So I'm going to explain the several kinds of control. So a concurrent control group is one chosen from the same population as a test group and treated in a defined way as a part of the same trial that studies the test treatment and over the same period of time. So types of control. So I'm going to explain the types of control. The first one is placebo concurrent control. So as you know, a placebo is a dummy treatment that appears as identical as possible to the test treatment with respect to physical characteristics such as color, weight, taste, and smell, but does not contain the test drug. Second one is no treatment concurrent control. This kind of control is rarely used, but for example, if we want to test the effect size of the placebo, the placebo effect, in that case, no treatment control group might be used. The third one is dose response concurrent control. So with that, in a randomized fixed dose response trial, subjects are randomized to one of several fixed dose groups. Next one is active concurrent control. So an active control trial is one in which an investigational drug is compared with a non-active drug. So active drug means its effect was shown in the historical trial. So if we use active control, we do not have ethical issues. Next one is external control. So an externally controlled trial compares a group of subjects receiving the test treatment with a group of patients external to the study, rather than an internal control group consisting of patients from the same population assigned to a different treatment. So external control can be a group of patients treated at an earlier time, which is called historical control, or a group treated during the same period of time, but in another setting. Okay, now next topic is when sometimes the clinical trial failed. So when a clinical trial failed, we need to think about why the trial failed. So reason might be, first one, the sample size is too small. Okay, so in that case, it is called the type 2 errors. Or we may have bias against test treatment. Okay, so that's why we need to minimize bias. Third one is poorly selected primary endpoint. Sometimes the primary endpoint failed, but secondary endpoint success. Okay. Or the fourth one is poorly defined patient population. Okay. Next one is truly insignificant hypothesis. And then the last one is safety issues. Okay. I think this is my last slide. Okay. Thank you.